Hello, thanks for coming. We did brief introductions before, but my name is Bragg. I'm a postdoc here working with Kristen Lyle at the Institute. And today I'm presenting on a little bit what I do here at the Institute, which is nutrition and autism research. So this presentation was created mostly to give summer scholars and those not super familiar with nutrition assessments a little primer on tools that we use to learn about people's diets, as well as a brief overview of what we do know about maternal nutrition and ASD development. So please feel free to jump in at any time with questions. Happy to chat more about any of this. So I'll take this opportunity again to introduce myself. I'm hoping this is helpful for summer scholars or maybe high school students who may be listening to this and are thinking about what kind of path they may want to take in the future. So I'm Bragg, I'm from Delaware, which is where I did my undergraduate degree in dietetics. And then I received my registered dietitian credential from UD as well. For those who aren't familiar, a registered dietitian is a nutrition professional. They've completed a 1-year internship program, which is transitioning now to a 2 years master's. And they are able to provide medical nutrition therapy. So they're the people you might see at the hospital or working one-on-one with clients. Whereas a nutritionist, which maybe you've heard this term, is a little bit different, and they don't have the same credential, but they can still work in nutrition fields as like school food service. You may see them at WIC or other nutrition locations. So happy to talk more about what an RD is, what a nutritionist is. After that, I went on to get my master's in human nutrition at University of Delaware, and then I decided it was time to move across the country, and I did my PhD at UC Davis. And I focused there on international maternal and infant nutrition. So my dissertation was on the nutrient choline, which is found in eggs and other foods, and how it relates to children's growth and development in Malawi in Africa. And while doing that PhD, I realized I was really interested in this question of how early life nutrition influences children's neurodevelopment. And that brought me back here. So I'm currently a postdoc researcher working with Kristen Lyle, looking at how maternal nutrition influences children's odds of ASD development. Summer scholars, other people listening, I'm happy to talk more about what a postdoc is, what a postdoc does, why I choose a postdoc if that's something you're interested in too. I'm sorry, our babysitter's coming in. So today I'll talk about 4 main nutrition assessment tools that we use: food diaries, 24-hour recalls, food frequency questionnaires, and a little bit about nutritional biomarkers. There are many others like weighed food records or food photo records, but they're not as common. I'm happy to talk more if that's something that you're thinking about using. So the first method we'll discuss is food diaries. This one's pretty straightforward. We just ask participants to keep a diary or a log of everything they eat or drink throughout the day. It's great because they don't need to try to remember anything. It's all in real time. So it helps prevent forgotten foods. Some food diaries are even on people's phones or their tablets, so they can take them with them wherever they go. But there are some challenges. It is a lot of work. It seems simple, but if you've ever had to record your diet, maybe on MyFitnessPal or another app, you realize that it's a lot of work throughout the day to write down not just the name of the food, but the brand, how you cooked it, how much you ate, what time you ate it. It can definitely be a source of burnout. And it also requires that participants are able to read and write, so it's not appropriate for all populations. Second, another challenge is that food diaries can influence food behaviors. So actually keeping a log of everything you eat throughout the day is a tool that dietitians use to help their clients manage their weight. When you're writing down everything you eat, you generally will eat less, and you'll eat maybe simpler or healthier foods just because of the act of logging. So this is really useful for clients of dietitians, but it's not great for researchers who are trying to get a realistic view of people's intake. And the third challenge is that this is a 1-day food record, and we eat differently on every day. So it misses out on some of this intra-individual variation, the variation within one person's normal eating habits. So for this, you might think about what your food log might look like on Super Bowl Sunday versus on the Monday after New Year's Eve, right? It's probably very different foods that you're eating. So to help overcome these challenges, we have a few best practices we use for food diaries. First, we need to train the respondents, train these participants. So this is how you fill out a food log. This is the information we're looking for. We encourage them also to be honest, to try to stick with what they would normally be eating. And often we'll add reminders that they can, to encourage them to stick with it. Because again, it is something that's easy to let fall by the wayside. Also, unless we have a representative sample and we only want a population average, we have to ask for multiple days of recall to overcome this challenge of inter-individual variation. So usually we'll ask for at least one weekday and at least one weekend day. But the number of recalls that you'll need varies depending on what nutrients or food you're interested in. So we eat some foods and nutrients every day, like grains or protein. So if you're interested in that, maybe you can do fewer recalls, but some foods and nutrients are eaten more episodically. So think seasonal foods, think foods we eat during celebrations. So it's going to take many recalls for them to actually be reported, which is something to think about. But this has to be a balance because the more recalls you ask for, the fewer people are willing to actually do them well. So, so you kind of have to think about what your goals are. To help reduce participant burden, you can also use the second tool, which is 24-hour recalls. In the 24-hour recalls, respondents recall everything they ate or drank within 24 hours. And this 24 hours can vary. Sometimes it's the 24 hours leading up to the interview. More often, it's the 24 hours ending with the previous midnight. So midnight to midnight on the last day is how we usually do it. Asking people to recall what they ate is actually kind of challenging, and performing a recall is an art. And the USDA recommends this multi-pass method as a way to help structure the 24-hour recall. So first thing we do is ask participants to go through, just go through their day and list everything they can think of that they ate or drank. Then we go through and ask about a list of forgotten foods or commonly forgotten foods to see if that triggers any memory. These are things like drinks, snacks, condiments, alcohol. Third, we ask for the time and the occasion. So the occasion just means, would you call it breakfast, lunch, dinner, or snack? And then the details pass. And this is the longest section. This is when we're asking about brands, cooking techniques, the amount, et cetera, et cetera, et cetera. And then finally, we check to see if they can remember anything else. So anything else during the day that you can recall? And 24 hours are actually my specialty. I performed over 500 of them during my master's program. And so I'd love to do a quick demo if anyone's interested in sharing what they ate yesterday. It would just be a mini one. So they're usually about 30 minutes, but we would just do a quick 5-minute one, maybe just doing breakfast or lunch. Would anyone be willing to share what they ate yesterday? If not, I can share and you can interview me. Nate, if you don't mind?
Yeah, I don't mind.
Okay, great. Then let me get out my little. So here I have, can you see the spreadsheet?
Yes.
Yes, I have food, time, location, brand, details, amount. So we'll start off with so the first thing you do in a real life, you would ask the person to come in or you'd be on the phone with them and you'd want to establish kind of a rapport. So you'd say, hi, I'm Megan. I'm a nutrition professional and I'm looking forward to talking with you today. I want to remind you that all foods are a mixture of good and bad, and there's absolutely, and everyone eats a mixture of good and bad foods. And the most important thing today is that you're honest and that you try your best to remember as well as you can what you ate yesterday. Generally, you would tell the person, no, this is just between you and me, no one else can hear. Obviously today, that's not going to be the case. And then we'd go ahead and get started. So Nate, could you tell me just a list of the names of foods that you ate yesterday? You could just do maybe the first eating occasion you had.
Okay, so I had what are they called? Frosted Flakes. That was the first thing I had yesterday. Okay, and then I could just keep going.
Yeah, keep going a little bit more.
Okay, so I had Frosted Flakes in the morning. Then I had a burger with fries. Next, I had like as a snack, I had some. They're called Parmesan crisps. I don't know if you guys know what those are, but I had those a little bit later on. And then for dinner, I had noodles, like microwave noodles.
Okay, so then we'll go through this forgotten probe. So any other drinks you can think of, like water
Oh,
or alcohol?
yeah. So let me see. So yesterday I think I had lemonade in the morning. I had Coca-Cola for lunch, like with my lunch. And then throughout the day, I was kind of drinking a Red Bull.
Great.
So yeah.
Great, and then we go through normally a list of all these forgotten foods and probe. Did you have any gum? Did you have any XYZ? Up next, and don't worry about it this time. I'm just looking at my list, time and occasion. So up next, we'd ask what time you're having everything. Let's just do the frosted flakes and burgers and fries for our purposes today. So what time do you think you had the frosted flakes?
I think like 8 a.m.
Okay, and what time do you think you had the burger and the fries?
That was at 1:50. I remember that.
And what would you call these occasions? I think you might have said breakfast for frosted flakes. Is that right?
Yeah, like breakfast and then the burger was lunch.
And now we're going to go into some of the details. So do you remember what brand the frosted flakes were? Were they store brand or regular?
Yeah, they were store brand. Trader Joe's store brand.
And did you have anything with them?
No, I just ate them dry.
And there's this part where we also point out we want to ask open-ended questions as much as we can. So it wasn't, did you have milk, or what kind of milk did you have? That one would be a no-no. What kind of milk did you have? Did you have skim milk with this? We want to make it as easy as possible for people to be as honest as possible. And we also want to maintain as non-judgmental and encouraging as facial tone and expression as possible. Which can be a challenge sometimes because people always like to surprise you with things that they ate. So, so that's another tip. And then we'll just finish up with, do you remember the amount of frosted flakes that you had? You could think in terms of cups, in terms of maybe a size bowl that you had.
I want to say like, yeah, like a small bowl is probably the best way to put it. Like, I can't think how much that would be like in like cups is the
How
problem.
about in the size of your fist? Can you think of bigger than the size of your fist or smaller?
Probably a little bit bigger than the size of my fist, a little bit bigger.
Maybe, you said 1.5 fists or less than that?
Like 1.5, yeah.
Okay, great. Thank you so much for practicing that with me. So another thing that we'll often do is to give them these food models or these measuring cups. So in real life, Nate, we would have some cups out for you or some bowls, and I would know the volume of each of the cups or bowls, and then you could tell me compared to that. Nate, how challenging or easy was it for you to remember what you ate yesterday?
It felt kind of challenging because after I went through the 3 meals, I'm like, I probably had some snacks somewhere in there, and I just couldn't remember.
Yeah, that's one of the main challenges of the 24-hour recall. You think that it's easy to remember what you ate literally 24 hours ago, but it is really challenging to remember not only what you ate, but how much you ate and all the details. So, so that's a very common response, is that it's hard to do. And this can lead to forgotten foods, things that we don't remember to disclose, and also to phantom foods, which are foods that you report eating, even though you actually didn't happen to eat it yesterday. Maybe you eat it every day, but you forgot, you missed that food yesterday. And even when people do remember what they eat, they may be subject to social desirability bias. And this bias is what leads us to underestimate foods that we consider healthy. So maybe say you ate a little bit less cake than you truly did, and to overestimate foods that we consider healthy. So that's when you're saying maybe you ate a little bit more fruit than you actually did. And this is why it's so important for the interviewer to be encouraging and non-judgmental. Hi, Diana, you have a question?
Yeah, I was wondering about if people take vitamins or nutritional supplements and things. Do you normally ask about that as well?
Yeah, we do. Yeah, usually that is kind of a separate supplement questionnaire that we'll add on. It's not always part of like a traditional 24-hour recall, but most of the time we do ask about it afterwards. Yeah, good question. And we'll also ask people, if they know why they take the supplements. And it's interesting that a good percent of people don't really remember exactly why they started taking a supplement if they've been taking it for a good amount of time, which I thought was kind of surprising. Another kind of downfall of this 24-hour recall is the same issue that we have with the food diary, which is that it's only one day. So again, we'll have to do multiple days of recall unless we're just looking for the population average. And then we talked about best practices. In real life, you'd want to have props like bowls, cups. They actually sell these really realistic looking little food models, kind of looks like kids' toys, but there's different sizes for different portions. Or sometimes when you're doing maybe an online or over the phone recall, we will send ahead this booklet that has pictures of what different portion sizes will look like. And we'll say, why don't you go to page 3 and that has a bunch of cups and bowls on it, and you can try to estimate how much you had from those. And the other best practice is to train and standardize interviewers. So again, making sure they're comfortable doing recalls. There are also computer-assisted 24-hour recalls that can be really useful. Kind of their drawback would be the inflexibility. So if someone's eating something that maybe isn't programmed into the recall, or they're not really sure how to describe it, then they'll miss out on that. So to overcome this need for having many days of recall, you can alternatively use a food frequency questionnaire. And this is the third tool we'll talk about. In food frequency questionnaires, or FFQs, participants report how often they consume food during a certain time frame. So it could be a week, it could be a year, it could be the first trimester of your last pregnancy, any time frame that you decide. And these questionnaires can be qualitative or semi-quantitative. So a qualitative questionnaire just asks about whether food was eaten and maybe how often. So on this example, over the past 12 months, how often did you drink carrot juice? It would just be this, this first question. So you get some idea of the number of times, but you don't get an idea of the amount. A semi-quantitative FFQ adds the second question, which is about how much did you drink when you drank carrot juice? And this allows us to get an estimate, a rough estimate of the estimated intake for certain foods and then eventually certain nutrients. And FFQs are nice because you can ask about these episodically consumed foods, these seasonal foods, as long as the FFQ is looking back over that season. So in the past year, how often did you eat, corn on the cob? I only eat it in the summer. That's when it's kind of fresh. However, this technique still is subject to social desirability bias, and it requires long-term recalls. So we're not just remembering what we had yesterday, we're remembering what we had, weeks or months ago, which is a big challenge. In addition, the food list must be appropriate. So it's a balance between wanting to decrease the amount of time. Some of these FFQs are hundreds of foods long, and it can lead to a lot of burnout for our participants. But you need to have enough foods on there that you're getting a good sense of their diet. If their staple food is yams and you forget to put yams on the FFQ, then you're going to be missing out on a lot of really important information. So best practices for FFQ, you want to ask about a reasonable time frame. So the farther back in time you go, the harder it is for the participants to accurately remember. If I asked you how many times per month did you drink orange juice in 2010? It would be very challenging for you to answer that question, but maybe not as challenging if I asked you last month about how many times you drank orange juice, right? Also, researchers have to have this deep understanding of local food practices, like I mentioned with this example of if you don't have their staple food on there, or if you don't have foods that are main sources of certain nutrients, then you won't be getting that information. It'll be completely missing from your data set. And if you are interested in a specific nutrient or food, there are validated FFQ screeners that you can use. For example, there's a choline one remember my interest is in choline for my dissertation, that asks about foods that are sources of choline. So those are options as well. I borrowed the slide from Bess Caswell at UC Davis, who is the postdoc in my lab during my PhD. And I like it because it shows the 3 assessment tools we've talked about so far on kind of this trade-off continuum. So on the left, you have this cheaper, faster, lower participant burden option, the FFQ. You know, it's something maybe if you have a large sample size, or if you want kind of more general information, that's a great option. And then as you go in intensity towards the food record, multiple days of food record, then it is more expensive and more time consuming, but it's also more accurate potentially. And so when you're starting a research project, you have to think about kind of where in this balance do I want to be? So eventually, each of these techniques will provide us with a list of foods that a person has eaten over a certain time. And if we're interested in foods or food groups, we can stop there. But if we're interested in food nutrient values, we need to do a conversion. So to convert foods to nutrient values, we usually get down to food portion per day. So if it's reported in months, you know, we divide by 30 or 30.5 days per month. And then we get down to grams, food portions, and then we convert to grams of food. And then we use nutrient values that are listed in food composition databases that provide the nutrient per gram. So I'll just show you one nutrient database. Here, this is the USDA's database. This is open to the public. Anyone can go to this link and put in any food that they're interested in. And they have different versions of the food. So if you're interested in egg whites, egg yolks, dried eggs, et cetera, you can click on it and it will give you the amounts per 100 gram or per egg, the amounts of water, energy, any nutrient that you're interested in, et cetera. So we use food data composition databases like this to get the nutrient values for each person. And eventually what we end up with is a file that, just let me pull it up, that looks like this, where we have all of the values of wealth of nutrients for each person. And we can use these data for statistical analysis, which you'll be hearing more about, I think, soon from Juliet. So stay tuned for kind of the next step after this. In the meantime, let me get the PowerPoint back up. So we've got this nutrient values from foods, right? And Juliet will talk more about how to analyze this. But each of these methods that we've talked about so far requires communication skills. And depending on the population, this can be a big challenge. Also, they are subjective measures, you know, they're coming through the lens of someone else. So our other option is objective nutritional biomarkers. So these are biological factors that objectively measure nutrient intake or status. For example, we can measure the amount of nitrogen in urine to understand how much protein you're eating. We could look at your plasma vitamin D to see your vitamin D status. And we even have some nutritional biomarkers for whole foods. So we can measure the amount of proline betaine in your plasma to get an idea of how much citrus fruit you've eaten. And given the challenges with the other tools I've talked about, it seems like biomarkers would be a no-brainer, but they also have their challenges. So a good biomarker is sensitive and specific to intake of the nutrient and food of interest, which means it increases and decreases when intake of that nutrient increases or decreases, and it doesn't change with any other nutrient. And in practice, this is really hard to find because nutrients are often correlated and metabolic processes are, you know, intermingled. And some nutrients are homeostatically controlled by their body. So for example, calcium is tightly regulated by our body. So the amount of calcium in our blood has pretty much nothing to do with the amount of calcium in our diet. So finding a good biomarker can be a big challenge. And when we do have a biomarker, specimen collection and lab analysis can be expensive because it needs to be accurate and reliable. You know, when you're doing this lab analysis, you need specialists who draw the blood, you need lab analysis, you need large machines, you need a cold chain to make sure that the blood stays cold, you know, while you're bringing it all the way from Africa to the lab. And participants have to be willing to part with their, you know, biological fluids, which is a challenge as well. So, so that's a major challenge. And then once you do have these specimens collected, biological levels of many biomarkers naturally vary with age and race, ethnicity, genetics, and even with things like fasting status. So you're asking people to come in after having not eaten for 8 hours, you know, how well do they adhere to this recommendation? So to work around these challenges, it's recommended that you use standardized methods for specimen collection and lab analysis, whenever you can, taking replicates of these. You should collect information on factors that could influence interpretation, like the time of day, the time since they last ate. And when possible, it's recommended to use multiple biomarkers or to use in conjunction with these traditional dietary assessment methods that we've discussed. So once we've used these tools, we try to connect them to outcomes and such as autism. And lucky for us, there's even more challenges coming up, which I think Juliet potentially we'll talk about, because some of the ways we work around these are using statistical techniques. But some of the challenges are that nutrients are correlated with each other. So and intakes are correlated with each other. So if you are eating a diet high in saturated fatty acids, you are likely to also be eating a diet high in sodium and potentially low in fiber. These are kind of dietary patterns that go together. And then these dietary patterns in themselves are also related to other healthy behaviors. So if you're eating this high fat, high sodium, low fiber diet, you may also be less physically active, or you may be a smoker. And these correlations can be teased apart with statistical methods and study design, but they are a challenge that we need to kind of think about. And then a more kind of interesting challenge to me is this interaction between nutrients with genes and environmental factors. And this is something that a lot of nutrition scientists are very interested in right now. And it's related to this idea of precision nutrition, which is kind of your own personalized nutrition effects. And despite all of these amazing challenges, nutrition epidemiology lives on. So, by employing careful study design, thoughtfully collecting data, and doing informed statistical analysis, we are able to find meaningful connections between nutrients and outcomes. For example, Zhang et al. Reviewed the association between prenatal dietary factors and children's odds of ASD, and found a list of nutrients that are associated with lower odds of ASD in the children. And some of these findings are using data from the Institute. And like, for example, polyunsaturated acid was Kristen's study. And not only are these nutrients directly related to odds of ASD, but maternal dietary factors can also attenuate the association between environmental toxins and odds of ASD, meaning that, having this exposure to air pollution can increase the odds of ASD. But if you're if you're taking prenatal vitamins, it may actually decrease that association and help attenuate it. And that was my paper that I worked on when I first entered the lab. So just briefly going to tell you about some upcoming projects we have in the lab related to nutrition. The first, I'm analyzing data from the early study. So I'm not sure if you've heard about this one yet, Nate, or if I bet Bridget will talk more about it, but it's an enriched risk cohort. So we're enrolling pregnant women who've already had a child with ASD. So they're more at risk for having for the subsequent child to be diagnosed. And we follow these women and their children out past 36 months or 3 years. And we collect data about the mom's pregnancy using 2 FFQs. So we do one FFQ that looks back on the first half of pregnancy, and another that looks back on the second half of pregnancy. And this is what led to the data set that I showed you a few minutes ago. So I'm using this data to see how maternal intake of choline and betaine during pregnancy affect the odds of ASD and autism traits in the offspring at 36 months. So choline and betaine are part of this 1 carbon cycle. Oops, maybe you can hear the baby. And they donate a methyl group or a 1 carbon group, which can be used for methylation of DNA, which turns genes on or off. And we know from previous studies that folate or folic acid over here towards the left of in this cycle is related to odds of autism. But no one's looked at these nutrients, which are also in this 1C cycle. So we'll be doing that. And so far, we're seeing a little bit of a signal come through with betaine in particular. We're also looking at physical activity patterns, and this is building on a master's student's work, Olivia Bessie's. And we're first kind of describing what are the physical activity patterns in women who have children with autism. We know that physical activity among pregnant women is many people are below the guidelines, and spoiler alert, it seems this population also is not meeting guidelines for physical activity during pregnancy. But we want to learn a little bit more about what this looks like for moms of children with disabilities. And then we're going to see how these patterns are associated with the subsequent child's odds of ASD. And then coming up, which is what I was brought to the lab to work on, we're going to be looking at which maternal dietary factors can modify the association between air pollutants and ASD. So kind of building on that review that I talked about to see if we can find individually modifiable factors that could help reduce the odds or the severity of ASD in the offspring. Oh, upcoming projects. So I hope this presentation gave you a kind of glimpse into the tools that I use every day and some of the challenges that we have in interpreting them, and also a little bit of information about how we use them specifically to autism research. But I'm happy to talk more with anyone, Nate or anyone else, about what I do and how I do it, and happy to take any questions now as well. Thanks.
Megan, I was wondering for your, with regard to this work and looking at these different nutrients and such, are you also looking at timing? So are you looking at different things, you know, kind of pre-pregnancy, you know, like at the time of, at the time of conception, first trimester, blah, 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 like you're looking at that factor as well?
Yeah, and it seems like timing is very important. So this relationship with folic acid and autism risk is specifically around the time of conception. And we're finding that really early pregnancy seems to be the time where nutrition is important. Mm-hmm. Yeah, and I should also note that some people are studying nutrition and autism in a completely different way, where they're looking at, the nutrition challenges of children with autism. The food options they're choosing and what kind of nutrient deficiencies they may be more at risk for. Some are even looking at, nutrition supplements as kind of adjuvant therapies, although I don't know that there's been too much traction there. But there are people looking at nutrition and autism in totally different ways than as a modifiable risk factor.
Very interesting.
Thanks. Saw something in the chat. Thanks. And it looks like Bridget will talk more about early next week. That's good.
I had a question really quick. So, okay. So you know how a lot of the time, they say that, okay, sorry, let me restart this. So because autism traits start to present sometimes a little bit later into childhood, around the age of 2, is it possible that maybe nutrition while the child is under the age of 2 and very young, do you think maybe that might have a factor into it, that kind of nutrition?
Yeah, potentially. That's something that we haven't explored very much specific to autism, but we know that this complementary feeding period where children are switching from breast milk onto family foods is a really important time of nutritional risk for a lot of children around the world. Because a lot of parents, well, mostly, so I'm taking this from the context of international nutrition, which is my background, but children are often weaned onto these foods that aren't specifically very nutrient rich. Things like, you know, corn porridge or rice porridge. And so some education or some speaking of the diets of young children can definitely be important for neurodevelopment in general, and I would guess for autism. But I don't know if I don't know that we've looked at like this complementary feeding period. It's a really great point. Yeah, a lot of brain formation happens earlier, and I would imagine that that's maybe more important in terms of timing, but there are, of course, brain regions that do continue and I think could definitely be influenced. Yeah, Diana?
Are you all using the early project for these studies that you're doing with Kristen, or are you using other data sets as well?
Yeah, we're using the early study for the choline and the physical activity, and we'll be using the early study and Nurses Health Study 3 for the air pollution modification study.
Okay.
Mm-hmm. So we're waiting on data from that one.
And how large is the sample from the Nurses Health Study 3?
You know, that's a good question, and I don't know the answer, but I know it's larger than early, but it's not an enriched risk cohort, so I'm not sure like what how many children with autism we might have.
Mm-hmm.
But we're also using for the choline data, for the choline analysis, we're also using Marble's data, which is a similar enriched cohort over in California. And that also that's going to increase our sample size to around 300 mother-children dyads. So a modest sample. Hopefully we can see some interesting things.
And what foods carry choline?
Mostly animal source foods. So eggs are a really rich source. Also fish, poultry, things like that.
Okay.
Yeah.
So it's not too rare then. It's not too rare.
It's not too rare. And you know, one of the interesting things about choline is it's recommended to be put in prenatal vitamins, but it's really bulky. So it's not in any prenatal vitamins that you can really find on the market. Well, I guess I shouldn't say any, but it's not in the common ones. It's not in ones that most people are taking unless they're thinking about choline. So it's kind of an interesting nutrient, and I'm wondering in the future if that's going to change, if people are going to make it work in vitamins.
It's really interesting.
Thanks. Yeah, thanks for listening, guys. I really appreciate the chance to.
And very challenging too. So yeah, I'm really, it's my hat's off to both you and Kristen for pursuing it. This is not an easy exposure to track.
No. Yeah. But a lot of work to be done. It's kind of an open field, so that's nice.
Mm-hmm.